And also thank you to the organisers, BRCCH, for inviting me. It was a pleasure to join the other two speakers on this important topic when we think about the challenges for malaria control and elimination. So I have the pleasure to speak to you about data analytics and modelling today, and particularly in the support of the development of new interventions. So as Simon introduced malaria and the life cycle, today I want to talk to you about the challenges we have in continuing with burden reduction and preventing malaria. So over the last 20 years, we've seen significant malaria burden reductions, and this is a huge achievement. But unfortunately, now we're seeing significant changes to this burden reductions. It's stalling, and in some cases, actually regressing. And in many cases, we've seen an uptick in cases, particularly in high burden countries, which is a consequence of COVID, but many other aspects, including resistance, funding gaps, and accessing children that are not getting malaria prevention. The challenges we have moving forward are quite significant. This is increasing insecticide resistance, drug resistance. We also see that we need tailored interventions and targeting interventions in high burden, but also in urban malaria. We also have challenges. It's not moving for me. We have unequitable access to treatment and interventions, but we've also unfortunately seen the invasion of Anopheles stevensii mosquitoes into the Horn of Africa. This is a very efficient mosquito transmitting malaria, which most of our vector control doesn't remain effective. And Simon also alluded to it when he talked about when we're preventing infections, we're seeing changing immunity patterns, which are going to bring quite some challenges over the next 5 to 10 years. So if we're preventing infections in children, what does this mean for anti-disease immunity? And so on, which has a consequence for us to monitor success. We can't interpret our disease statistics the same way moving forward. I apologize to everyone online. I have to turn back just to check my slides. I can't see them on the machine. So Simon introduced the malaria life cycle. So prevention actually comes in many forms. We can prevent malaria in the mosquito, we can halt transmission from the mosquito to the human, or we can tackle the parasite burden in the human host and prevent sickness and illness in children, or prevent onward transmission. So the prevention we have is in the mosquito. This first slide that maybe you can still see was that we have many places to intervene for malaria prevention. On the vector side, we have quite some tools available, which include insecticide-treated nets, insecticide residual spraying, but also forthcoming is gene drive and attractive targeted sugar baits. On the human side, we can combat malaria, as Simon suggested, at the liver stage, the blood stage, and also transmission blocking. Of course, anti-disease is in the blood stage. Okay, so let's take a step back. We're talking about a recap. Malaria is complex, and all the tools we have against it are quite imperfect. And there are many points in the parasite and vector life cycle where we can have points of evolution. The tools that we have today are effective against the vector and also the parasite, but we have new tools for the vector control coming up, which include gene drive and hopefully attractive targeted sugar baits. On the human side, we're going to be talking about some of the malaria prevention on the human side, which means preventing infection, as Simon said, with the anti-CSP or liver stage immunity or drugs. So this can also be vaccines or monoclonal antibodies when we talk about immunity. At the blood stage or against the clinical disease, we have drugs and chemoprevention, but of course we also have forthcoming vaccines potentially. Transmission blocking, we also have drugs and vaccines. So our toolkit to meet these challenges for malaria control and elimination today and over the next 10 years are quite varied. We know in principle what to do: clear infections, reduce transmission, and prevent reintroduction. But as Simon alluded to, there are many other challenges, including what happens if we really delay acquisition to immunity against disease. So one other tool that we also have on top of the tools I've just mentioned include modeling and analytics. And today I'm happy to talk to you about how some of that might help us think through these new novel prevention tools. So you may be asking yourself, how does modeling work? How can it really support the development and the R&D, the research and development of a new medical intervention? So I'd like you to take a step back and imagine that you're Simon or another product developer who has an antigen and a vaccine. You've seen some great preclinical results, and you're wondering how can this product of a clinical disease and mortality in children, maybe it's a monoclonal or a drug, but you've got significant preclinical evidence to warrant thinking about phase 2 clinical studies and early human studies. But already at development, you have to consider who your patient is or who your target population for prevention is. And you should already start thinking about the community as well, about acceptance, how we might deliver that tool, is it seasonal or perennial. And then on top of that, you also need to consider the ecosystem you're in. What are the current interventions that I've just mentioned that already exist? What's the funding mechanism? Who's going to buy the product? Is it the community? Is it donor funding? Is it individual patients looking for their own prevention? And that's in context also of the current disease dynamics ongoing and potentially changing burden and population immunity. So you may have a product, you think you can stop malaria, the invasion of the monocyte into the blood stage, or another CSP antigen, but you still have far too many questions to answer. And then you also need to really, on top of that, think about what is my goal? If I'm going to get this approved at WHO, what are the efficacy targets? What are the lives saved that would enable WHO to recommend this as a broader use in the population? So you're balancing all these questions to get research and development investment. And in the end, you're really asking, is my intervention got the properties to achieve a public health goal? How will I use it? And you're balancing those priorities along with your partners. And we have good news. This is where modeling can kind of help you do this instead of heuristics and just in conversation. Modeling can help put some quantification around these questions, and not just disease models thinking about the total impact you might have. You can triangulate it with economic analysis and the intervention landscape and look at how it might behave synergistically with other interventions to be impactful. So today I'm going to talk to you about 2 use cases of modeling to support R&D development. The first is really more the traditional type analysis where you take a product and estimate the potential public health impact. So how many lives might you save with that new vaccine, for example? And the second is the role of modeling to kind of flip that question and say, what are the characteristics of a new vaccine to be successful? And how should I use it, and with what combinations of interventions? So let me talk you through those use cases. And before I do so, I'd like to just point out that malaria modeling has a very strong history. So it's not just something new that we're doing, which many people may have been exposed to infectious disease modeling during COVID. There's a long history of malaria modeling that goes right back to the end of the 1800s, 1900s with Sir Ronald Ross, when he used mathematical modeling to really communicate the disease dynamics and how we might intervene. So modeling was no more than putting a framework, a conceptual framework around against your understanding, current understandings, and then try to see where to intervene to have largest impact. You'll also note I put Hilda Hudson here. So around 1916, she also wrote several books with Ronald Ross on pathometry. And I bring this up because that was the very beginning of what we know as the effective reproductive number. So using thresholds, the concepts of thresholds to describe when a disease increases or what a threshold is to actually decrease that disease. So the types of models have moved quite a lot in that time. They've become more complex and detailed. For example, there are individual-based models of malaria. For example, something that we have at the Swiss Tropical and Public Health Institute is a model called Open Malaria. This type of model, and there are several others out there, try to capture the disease with the precision required for the question that we're asking. The individual-based models allow us to look at some stochasticity and variation in the disease, but what we do in essence is capture what happens within a human host, the parasite growth, immunity acquisition, and then also how disease clinical symptoms might progress. So there's age patterns of disease, immunity acquisition, we've got acquired immunity, and we've also got immunity decay. And that all those models within the host are linked to a vector model, so the mosquito model, the life cycle. The models out there are all calibrated to historical data. So we have these models with sufficient detail to ask questions about current interventions. And some of these models also include detail that allow us to model things about our drugs or our monoclonal antibodies or our vaccines in sufficient detail to talk about research and development. So we've increasingly used models that capture explicit parasite dynamic growth models. We can track different genotypes, so we can look at drug resistance, and we can also include immunity models or pharmacology models of drugs, for example, which are very explicit, so we can model parameters that a drug developer may be trying to optimize. And it allows us to look at a lot of questions, including on drug resistance, that support understanding when we might replace our current first-line treatment with a new drug. So also supporting research and development advocacy. So the two use cases I discussed today, in principle, we take one of those models that I just showed that capture the dynamics of malaria within a human host and between the mosquito and back again. And we can, for the first use case, we can look at estimating the potential public health impact of a vaccine. These models that take into account they take our known understandings of malaria, data, biological, epidemiological, they inform the models. If we want to look at the potential impact of a vaccine, for example, then we also input our current knowledge, the efficacy and maybe how long the antibody kinetics lasts, for example, estimated from a clinical study, and we estimate potential public health impact. Everyone, I think, will be used to this. The models that were used during the COVID for the vaccine rollouts did this type of scenario analysis. What if we roll out a vaccine at a certain rate? How many lives might we save? The second approach that we're talking about today is where we flip the question and we reverse the analysis. And now we ask ourselves with our new vaccine, do we need to be better than the current vaccine? Do we need to be better than an existing intervention? So we already have some estimate or target population health target that we're looking for. For example, it could be 80% of clinical cases averted within a season to be superior or at least non-inferior to seasonal malaria chemoprevention, for example. So if we set a target, what we can do now is use these models, flip the analysis, and search for the parameter space of the new vaccine or drug that might be able to achieve that target. So what this means is we're then getting some quantification around the key performance characteristics of a vaccine or a drug that would be able to achieve our target. And this type of analysis can then inform guidance documents for product developers, for example, a pharmaceutical, knowing now what they kind of need to achieve to reduce malaria burden. So let's go through some examples. Simon mentioned the antibody kinetics of RTS S. This is a very good example of where data analytics and modeling was used to try and understand what was observed within a human host and then what was observed at clinical trials. So this is work that was done at Imperial College, where hopefully they had the right immune correlate. We'll look at antibody titers and modeling that type of decay, correlating it with the dose-response relationship to match the efficacy against clinical disease and backtracking the efficacy against infection that they observed. This is a nice, neat use of modeling where you take individual level data, you use dose-response curves, and you look at your predictions in the clinical trial. They further then looked at other immune correlates or other immune aspects that Simon also mentioned. So they've also looked at avidity. So rather than using one aspect of immunity, they've also looked at avidity and immune titers, which also allows you to answer other questions about if you could increase that avidity, would you then also get to maybe longer immune kinetics, for example. The second example is now the traditional sense of modeling, which is predicting the public health impact of a new intervention. There was an example following the RTS S trials that Simon introduced, and this is also ongoing for the R21 vaccine and antisporozoite vaccine, where modeling is used to estimate what might happen beyond the clinical studies. So here in 2015 and continuing through pilot implementations in 2018 and up to last year, clinical trial data and implementation data was used by the modeling groups to inform their models. And then they did scenario analysis beyond the trial studies to work out how many lives might be saved in other settings, how cost effective might it be, and then to look at questions beyond those utilized. For example, originally the vaccine targeted children. Modeling was done to see what might happen if you use it as a seasonal use case, for example. This type of analysis is not only done in malaria. It's also done, for example, dengue vaccines and COVID vaccines. This type of modeling was used as one part of the evidence that formed the package for recommendations on vaccines, and also for Gavi, the Vaccine Alliance investment in malaria vaccines. And moving forward, this modeling is also useful for a new intervention to decide how to implement it. For example, programmatic decisions, and moving forward also how equitable access in a dose-constrained environment that we have with RTS S. So I think Evelyn will talk to the implementation, lessons learned of RTS S in Ghana. There is now more questions what happens with the small number of doses with RTS S, and modeling is informing some of the equitable framework in that. So the last use case is to talk about this when you keep the target population and public health goal in focus to define the characteristics of a brand new intervention that may only be in preclinical studies and not yet in clinical. So I pointed out we can use modeling to say what is our target, and then reverse the analysis and define the efficacy, duration, or settings, which means we would meet that target. So working with the Bill and Melinda Gates Foundation, we currently at the Swiss Tropical and Public Health Institute have a project to kind of give some quantitative evidence towards the key characteristics in three of their investment documents and product documents. These investments or product documents, they're called target product profiles, are primarily around replacing current seasonal interventions, but they're exploring other use cases such as perennial use of prevention, medical prevention. So we're looking at seasonal malaria chemoprophylaxis, which is a mainstay intervention where we give 3 to 5 rounds of a chemoprophylaxis drug to children to prevent incidence and onward severe disease. Then looking at whether we can use long-acting injectables instead of coming up to 5 times a year. It could be delivered to children once or twice. Simon alluded to monoclonal antibodies, which is an exciting space at the moment, which there are some trials ongoing. So we're working with the foundation to understand the performance characteristics of a monoclonal to be able to come in once, maximum twice a year to prevent malaria in perennial and seasonal settings. And then, of course, what's the longer term objective for malaria vaccines? Used as a multi-seasonal vaccine or as elimination. So this work has been led by Lydia Brennett-Meyer, Nariman Nakab, and Josephine in the audience, and also Sherry Kelly with myself. So I'm going to talk you through what we're doing and a few key results before I hand over to Evelyn. So the idea of this project, yes, we can provide some key performance characteristics, but the idea is not to do it once. The idea is to work with our partners, for example, NIH with monoclonal antibodies, to incorporate clinical evidence as it comes on board and to refine the characteristics, refine the settings we might use those products as we get more information about the responses in early human studies. It is a cyclical piece of work guided by experts and the clinical evidence. It's not just done once and in isolation. And the hope is that all stakeholders are on board with what's going on in the target product profile guidance documents. So we've got quite some partners who've actually informed the work that we're doing. It's their questions that we're answering. And a few of these we're working quite closely with on the vaccines, the monoclonals, and drug development. So the first example, which is now at completion, is second generation seasonal malaria chemoprevention. We were asked by the Bill and Melinda Gates Foundation to work with WHO and Medicines for Malaria Venture to really look at what characteristics of a drug to replace our current sulfadoxine, pyrimethamine, amidiaquine drug that is used as a seasonal malaria chemoprophylaxis, chemoprevention drug. They're developing new drugs, but they're also looking at repurposing and recombining existing drugs. So the questions asked to us were, what does that new drug need to look like to be equivalent to or better than sulfadoxine, pyrimethamine? And also, what would it need to look like in the face of resistance? So the type of analysis that was conducted was similar to the framework I showed you before. We took the targets given to us, for example, 80% clinical incidence reduction, and worked backwards to look at the efficacy and duration requirements. What Lydia did was, though, because we don't know what the future drugs will look like, we modeled different modes of actions. So this is where we have the pharmacological models and the parasite models. We can actually play with, should it be blood stage or dominant liver stage, for example. Lydia also did a trial validations. She compared her results against existing trial and observational data on SMC. So she found what you would expect, that coverage is a key driver of the impact we'll see. And of course, that lasting protection between rounds is crucial. We would all say that, but what she's able to do is also put some quantification to the elimination half-life needed, and also to really give context around how to use the drug when we first get results of early clinical studies, that the duration of that elimination half-life may determine how we deploy a new chemoprevention drug. And of course, as you can imagine, it also gives advocacy to really looking at the formulation of your drug to ensure high access and uptake. So this is going away, if possible, one dose, and also to look at efficacy versus duration, depending on your target. So, for example, she was able to show very concretely that we need an elimination half-life of around 10 days to ensure we meet our target of 80% in most settings of clinical cases averted. What's nice about this type of modeling is it isn't just the result, it's the conversation that goes around it. This work has also prompted really strong discussions about the fact that we don't know or understand enough about the mode of action of our current standard of care. And so we really need to understand sulfadoxine pyrimethamine prophylactic period today. So in 5 years, we're not concerned about any recommendations. We have enough to compare the two products against at a final recommendation or funding decision. And I'm going to end on vaccines because we had a nice introduction from Simon about vaccines. And the questions that Simon came up with are the types of questions we're looking at answering. I will say, though, we're not dealing with the difficulty of how to make vaccines last longer. We're happy that Simon's doing that. But what we're trying to do is put what can you gain by getting an extra 6 months, 12 months longevity above that target that Simon showed with the antibody kinetics. And so that's work is being led by Josephine. She's done some work on multi-seasonal vaccines. What do we get if we give children one vaccine with a slightly longer duration? What does it buy us in the second season? I'm not going to show that today. I'm just going to give you the big picture of let's go from today's protection of children to elimination vaccines. What could a vaccine look like to accelerate? And so Simon talked about this need to get a longer duration. So what we're doing is using this framework with Josephine to try and understand what duration do we need versus the trade-off of the efficacy for different use cases. If it's elimination or accelerating elimination, and in what context, where and with which interventions, is to quantify this trade-off between efficacy and duration. So as Simon said, there's quite some rapid developments over the last couple of years in vaccines and quite some exciting information coming out. And so our first initial work is focusing on CSP liver stage vaccines as preventing infection for these elimination, looking at exactly what Simon pointed out, not having a booster every year, maybe once or twice, or maybe no booster at all after the primary series. What is the value of that type of vaccine for different durations? But we are also then looking potentially, hopefully with Simon, at multi-antigen vaccines as well. So this is not an easy project because there are lots of steps in the process of defining your health goal. You need to define what success looks like. How could a vaccine be used as an accelerator for elimination? So here we have a lot of endpoints to discuss. And these are endpoints for your public health goal, but then we have to think about endpoints for the clinical trials as well. So the type of endpoints we're looking at are rate of resurgence if there's introduction, example, what maximum prevalence reduction is required for a primary series, and so on. What this looks like, though, is really we're doing kind of scenario analysis as well of different settings and trying to define durations of different vaccines that would allow us not to have a strong resurgence. And by that, the settings also include a pre-vaccination campaign where you assume children are protected either by the same vaccine or chemo prevention. There are other interventions ongoing. And then you look at deploying a primary series with or without a booster. And then you define which of the duration and efficacy target, what are the efficacy and duration ranges that might meet our target of what we define as success. And success is something we're working with our partners on for defining an accelerator vaccine. So basically watch this space, and we hope to discuss with Simon a bit more about multi-antigen vaccines as well. And so with that, I hope I've given you a flavor of the role of modeling for today, looking at impact, and for tomorrow for R&D investment. There are a lot of people to thank, including my partners sitting here, those that have contributed to a lot of this work. I've been at the Swiss TPH for a while, so there's so many people that have contributed to the models, and all of our partners working on this project, which include the Bill and Melinda Gates Foundation and the Gates MRI. And of course, I need to acknowledge all those that have worked on the extensive RTS S trials over the years and their funders. And thank you very much.